Hi there! Welcome to MCDB 427 Molecular Biology online mini course at the University of Michigan. My name is Yu Wei. Today we're going to talk about how eukaryotic enhancers activate transcriptions at a distance. This video focuses on figure 12.18, 19, and 20 in the book Molecular Biology, 5th edition, written by Weaver. As we all know, eukaryotes have enhancers that could activate transcriptions, even at a reasonably remote distance. However, the mechanism had been unknown. A more specific question is that scientists wanted to know whether an enhancer can work if it's physically located near a promoter but not on the same DNA. There were four most reasonable hypotheses as to how enhancers could simulate transcription from a distance, presented in figure 12.18. In the following figures, the blue box indicates the enhancer, the green circle is the activator, and the orange box is the promoter. The first mechanism is coiling. When an activator binds to an enhancer, it could potentially change the topology or shape of the DNA and even cause supercoiling. At the same time, this change opens up the promoter region for the general transcription factors. The second mechanism is sliding. After an activator binds to an enhancer, it could slide down the DNA until it hits the promoter. By directly contacting the promoter, the activator could activate transcription. So far, we can notice that the first two mechanisms require the enhancer and the promoter to be on the same DNA. Otherwise, coiling on one DNA molecule has little effects on another DNA, and the activator wouldn't be able to slide down the same DNA from the enhancer to the promoter. The third mechanism is looping. The activator could bind to the enhancer, loop out the DNA in between, and interact with the protein at the promoter to activate transcription. The essence of this model is not the looping itself, but interaction between the proteins bound to the two sides. Therefore, in theory, it should still work if the enhancer and the promoter are on two different DNA molecules, as long as the molecules were tethered together so that they could not float apart. The fourth mechanism is looping and tracking. The activator would bind to both enhancer and a downstream DNA segment to form a loop. And then the activator tracks towards the promoter along the DNA by making the loop bigger. This model requires both protein-protein interactions and activator tracking on one DNA molecule. So, how do we know which one is the actual mechanism happening in eukaryotes? Dunaway and Droge performed an experiment in 1989 and confirmed that the third model is the right one. What did they do? How did they conduct the experiment and come to this conclusion? Let's take a look at their experiment setup. First of all, they made a catenane, as shown in figure 12.19. In order to show that the protein-protein interaction doesn't need the enhancer and the promoter to be on the same DNA molecule, but still requires them to be tethered together, Dunaway and Droge constructed a plasmid PPD40E. It has an enhancer on one side, a RNA minigene on the other side, about 3.7 kilobase pairs apart, with a promoter, and lamina phage integrase sites, ATTP and ATTB in between. These plasmids were then recombined in vitro using phage lamina integrase and formed the catenane. Therefore, we have the enhancer on one DNA molecule and the promoter on the other, and these two molecules are still linked together. If this catenane could be proven to enhance transcription, then we could reject all other three models, because they all require enhancer and the promoter sequences to be on the same DNA molecule or requires DNA tracking. They've also constructed a series of plasmids under two groups. The first group is reference plasmids. They are plasmids with a 52 nucleotide RNA minigene only, but no enhancer. This serves as a control because it has the same promoter as our experimental plasmids. We will use this to normalize any variations in our samples. The second group is testing plasmids, including plasmids with a 40 nucleotide RNA minigene, which has the same promoter as the reference plasmids and no enhancer. Transcription from this plasmid should be identical to the reference plasmids. Plasmids with a 40 nucleotide minigene and the enhancer close to the promoter. This is used to determine the increase in transcription we see when the enhancer is in its optimal position. Plasmids with a 40 nucleotide minigene and the enhancer 3.7 kilobase pairs apart from the promoter. This is to determine how much transcription do we get when we move the enhancer away from the promoter. And the catenane, the one we really want to assay. It is used to test if there will still be enhanced transcription when the enhancer is physically close but not on the same DNA molecule. A plasmid containing the 52 nucleotide mini-gene, we call it Cy52. A plasmid containing the 40 nucleotide mini-gene, we call it Cy40. These plasmids were later injected into xenopus oocytes with equal molar amount of the references with the tested plasmids, and they measure their transcription by quantitative S1 mapping, which uses a single-stranded labeled probe to hybridize two transcripts, digest with S1 nucleus to degrade single-stranded or DNA or RNA, denature, and then the length of the probe is determined by high-resolution gel electrophoresis. One thing worth mentioning is that since we are making two different types of probes for the experiment, one for the 40 nucleotide transcript and one for the 52 nucleotide transcript, even though we're using the same promoter and the same environment as the reference plasmid, the radioactivity of these two types of probes may vary. Therefore, we might see bands with different strength on the gel. This is one thing that we need to take into account while controlling the measurement in experiments, by normalizing each data to the one with just the promoter. Another thing needs to be considered is the internal control. As we might have encountered, different oocytes could possibly influence the transcription activities. Therefore, we need an internal control for variations in the injection technique and for the recovery of RNA from the injected oocytes. For every experiment, we co-injected the two types of plasmids in equal amount to provide the control. One is a modified version of Cy40, and the other is Cy52 with the same promoter but no enhancer. This serves as an internal control so that we will be able to compare the transcription activities among different experiment groups by taking the ratio of Cy40 S1 product and Cy52 S1 product. Finally, it's time for the experimental results. Dunaway and Droge did the following 6 groups of sub-experiments, and the S1 mapping results are shown in this figure, with the numbers corresponding to each group. In the first group, they co-injected the Cy40 without enhancer and the same amount of Cy52 into the same oocyte, and measured the transcription activities of each. In theory, we would expect transcription from these two plasmids to be identical because they have the same promoter and only differ by the mini-gene. But we see a 4-fold hotness of the Cy52 in lane 1b over lane 1a. This is because the Cy52 probe is 4 times more radioactive than the Cy40 probe. There is really the same amount of mRNA transcribed from each promoter. Since we always wanted to ask how much transcription from our test relative to our reference occurs under each condition, we always have to take the difference in probe radioactivity into account and normalize our data. In the second group, in order to test whether their enhancer really enhanced the transcription, they co-injected a Cy40 with an enhancer close to the promoter together with the Cy52. And we see a lot darker band in lane 2A. It was said that this was a 17.5-fold transcriptional advantage on plasmids with the enhancers over those with mini-genes only. Taking into account the probe hotness difference, 17.5 divided by 0.25 is 70. Therefore, we could see that the existence of enhancers right next to the promoter introduced a 70-fold enhancement on the transcription. In the third group, to see if the enhancer could be moved, they co-injected a Cy40 with an enhancer about 3.7 kilobase pairs away from the promoter, together with the Cy52. It was said that this was a 1.5-fold greater transcriptional activity than the promoter alone. Taking into account the probe hotness difference, 1.5 divided by 0.25 is 6. The existence of enhancers a bit far away from the promoter still introduced a certain amount of enhancement on the transcription, even though it was not as good as the previous one with the 70-fold. The fourth group is the most important group. To test their hypothesis and see whether the enhancer can work if it is physically located near a promoter but not on the same molecule. They co-injected the purified enhancer promoter Catenane, Cy40, together with the Cy52. And clearly, lane 4A has a lot darker band than lane 4B. It was said that this was a 2-fold higher transcriptional activity than the normal plasmids. Taking into account the probe hotness difference, 2 divided by 0.25 is 8. The Catenane can enhance the transcription by 8 folds. This means that the promoter and the enhancers don't have to be on the same molecule in order for the enhancers to work. Before we dive into conclusion, let's take a look at the other two groups of experiments and exclude some other possibilities. To exclude the possibility that enhancers can work on a separate plasmid that's not physically linked to the plasmid with the promoter. In group 5, they injected plasmids with the enhancer only, plasmids with Cy40 with the mini-gene only, and the Cy52. We found that the transcription ratio of length 5A over 5B is the same as 1A over 1B. This means that if the enhancers and the promoters are on separate plasmids, the enhancers cannot activate transcriptions. Also, recall that when they were trying to make catenanes, they did not get 100% catenate due to the reversible reaction. They were worried that the low percentage of uncatenated plasmid would give them the enhanced transcription they saw in group 4. To exclude this possibility, they added about 5% of the PPD40E to group 5 setup. And the result is that we don't see a significant change on the ratio of length 6A over 6B. This confirmed that the transcriptional activation observed with the catenated enhancer was not due to a small amount of unrecombined PPD40E. Up till this moment, we could make some confident conclusions. The first conclusion, the enhancer doesn't have to be on the same DNA molecule as the promoter in order to activate the transcription. Secondly, however, the enhancer does need to be close enough to the promoter to be able to approach it so that the proteins bound to the enhancer and promoter can interact. Overall, we confirmed that Model 3 was the most reasonable model to illustrate the mechanism of eukaryotic transcriptional activation by enhancers at a distance. That's all I have for figure 12.18, 19, and 20. Hope this is helpful. Thanks for watching and go Blue!